Today, we're going to talk about the original DNA fingerprint. It's got a mouthful of a name, Restriction Fragment Length Polymorphism, or just RFLP. But what it did was actually pretty simple and totally revolutionary. This was the first time we could look at two different DNA samples and say, with real confidence, hey, these are not from the same person. It literally changed everything.
So, let's start with a question you've probably heard on every crime show, right? How in the world can DNA pinpoint one specific person when we are all genetically 99.9% identical? It seems impossible, but the answer is what makes this tech so incredibly cool.
The secret? Well, it's hiding in that tiny leftover 0.1%. Sprinkled throughout our DNA are these little variations called polymorphisms. You can think of them like tiny single-letter typos in the massive instruction book of you. And those little typos are exactly what make your DNA unique. RFLP was a technique built specifically to hunt for them.
All right, so how do you actually spot these incredibly small differences? I mean, you can't just put DNA under a microscope and see them. You need a special tool. And in this first part, we're going to see that this tool is basically a pair of molecular scissors.
These molecular scissors are actually proteins called restriction enzymes, and they are unbelievably precise. Each one is programmed to find one and only one specific sequence of genetic letters. That's its recognition site. And then, snip, it cuts the DNA right there. It's actually a trick bacteria came up with to chop up invading viruses. But scientists figured out how to borrow these little guys for our own detective work.
And here is where the magic happens. What if one of those little genetic typos, a polymorphism, lands right in the middle of a cutting site? Well, suddenly the molecular scissors don't recognize it anymore. They just float on by. So in one person's DNA, you get a cut, but in another person's DNA with that tiny typo, you don't. And that one small difference is the entire basis for this technique. So here's the bottom line. That tiny, single-letter difference means you end up with DNA fragments of different sizes. And that difference in fragment length, the length polymorphism, is something we can actually see and measure in the lab. This is the fingerprint we've been looking for.
Okay, so that's the theory. But how does it actually play out in a real lab? Section 2 is going to walk us through the step-by-step blueprint for RFLP analysis. Now, this process was incredibly powerful, but man, was it slow and complicated. Let's walk through it. First up, you take your DNA and you chop it all up with those restriction enzymes. Next, you sort all those little fragments by size using a technique called gel electrophoresis, basically running an electric current through a gel. After that, you have to transfer that DNA pattern onto a special membrane to lock it in place. That's called a southern blot. And finally, you wash it with a radioactive probe, which is a tiny piece of DNA designed to stick only to the fragment you're interested in, making your unique pattern of bands light up on an X-ray film.
So, once scientists had this method figured out, a whole new world of genetic analysis just blew wide open. Let's take a look at how people actually started to use and read these new DNA fingerprints.
The applications hit fast, and they were game changers. For the very first time, forensics could create a genetic fingerprint to link a suspect to a crime scene. Paternity tests became almost foolproof. But maybe the biggest deal was how it let us hunt for diseases. Researchers could follow a specific RFLP pattern through a family and see if it always showed up with a disease like sickle cell anemia. If it did, they knew the gene causing the disease had to be right there on the chromosome, next to that RFLP marker. It was a massive leap forward in mapping our own genetic code.
And you know, this incredible power of identification wasn't just for people. The same idea is still really useful in other fields, like agriculture. Let me show you a really cool example of how RFLP can tell different plants apart, even when they look exactly the same.
What you're looking at here is a snippet of genetic code from 3 species of amaranth, a really valuable grain. Now, just by looking at the plants, you might not be able to tell them apart. But look closely at the gene sequence. See that? In one species, A. cruentus, there's a T right where the other two have a C. That's a classic single-letter polymorphism. And that one little letter change makes all the difference. It turns out that it creates a perfect recognition site for a restriction enzyme called MSEI. So when scientists use that enzyme, it cuts the DNA from A. cruentus, but it completely ignores the DNA from the other 2 species. What you get is a clean, reliable, species-specific RFLP pattern. It's a positive ID that you could never ever get just by looking at the plant.
So if this technology was so powerful, why don't you hear about it in every crime lab today?
Well, that brings us to our final section, understanding RFLP's legacy in the modern world.
So, yeah, why don't we use RFLP all the time? The simple answer is that technology just got way, way better. I mean, look at this comparison. RFLP was slow. We're talking weeks to get a result. And it needed a pretty big, high-quality DNA sample, which you definitely don't always get at a crime scene. The newer kids on the block, like PCR and DNA sequencing, are just ridiculously fast. They can work with a microscopic amount of DNA, and they're way easier to automate. For most jobs today, they're just a much better tool.
But listen, just because it's been replaced doesn't mean it wasn't important. It was foundational. As the scientist Robert Williams put it way back in 1989, RFLP was the tech that triggered a race to map the entire human genome. It was the critical first step, the tool that showed us it was even possible to systematically map our own genes.
So when you think about RFLP, remember this. It might be history, but it's the foundation our entire genomic era is built on. It kicked open the door by proving we could read our own genetic code by focusing on our tiny differences. And that really leaves us with one big question. RFLP opened the first door. Now that our tools for reading DNA are getting even faster, cheaper, and more powerful, what incredible new doors are we about to unlock? What questions about who we are will we finally be able to answer?